Process of photosynthesis.
The chemical process through which green plants containing chlorophyll combine carbon dioxide and water in the presence of sunlight to prepare food in the form of glucose is called photosynthesis. Plants take in carbon dioxide and release oxygen through pores on their leaves called stomata. Singular, stoma. They take in water and minerals with the help of their roots. This carbon dioxide and water combine when energy from the sunlight trapped by chlorophyll reaches them. In the process, carbohydrates and oxygen are formed. Carbon dioxide plus water
in the presence of sunlight and chlorophyll
gives carbohydrates plus oxygen. Activity 1. To show that chlorophyll is necessary for photosynthesis. The leaves of a croton or coleus are partly green and partly non-green. Such leaves are called variegated leaves. Pluck a variegated leaf from a plant, perform the iodine test, and hold the leaf against a source of light. What do you observe? You will see only the green part of the leaf turns blue-black. This shows that only that part of the leaf which contains chlorophyll has prepared food. Activity 2. To show that carbon dioxide is necessary for photosynthesis. Take 2 potted plants and de-starch them by placing in a dark room for about 48 hours. Now place both of them in sunlight as shown in the figure. Cover the plants with bell jars. Put potassium hydroxide, KOH, inside the setup A. Use Vaseline to seal the bottom of the jars to ensure that no fresh air can enter the jars. After 6 to 8 hours, pluck 1 leaf each from both the plants and test them for starch. The leaf from setup A will not show positive starch test, whereas the leaf from setup B will show a positive starch test. This is because KOH was kept within setup A. KOH absorbs carbon dioxide, thereby preventing photosynthesis from taking place in setup A. Activity 3. To show that sunlight is necessary for photosynthesis. De-starch a potted plant by keeping it inside a dark room for 2 days. This will ensure that there is no starch present in its leaves. Now cover half a leaf, front and back, with black paper. Keep the plant in sunlight for 6 to 8 hours. Pluck the covered leaf from plant, uncover it, and perform the iodine test. What do you see? You will observe that the covered half of the leaf does not turn blue-black while the other half does. This shows that no starch was produced in the covered part of the leaf. This is because no sunlight was available to the leaf to carry out photosynthesis. Activity 4. To study the effect of sunlight on the rate of photosynthesis. Fill 3/4 of a beaker with clean water. Submerge an aquatic plant like hydrilla in it. Invert a glass funnel on top of the plant as shown in the figure. Fill a test tube with clean water and invert it on the stem of the funnel. After some time, you can notice that some bubbles are coming out from the glass funnel. This is due to photosynthesis in plant. The gas bubbles represent oxygen gas. Keep this setup in bright sunlight and then in shade. What do you observe? The number of oxygen bubbles is higher when the hydrilla plant is exposed to bright sunlight compared to when it is in shade. Activity 5. To grow fungi on a slice of bread. Moisten a slice of bread and keep it in a warm place for 2 to 3 days. You will notice some colorful fluffy patches growing on it. These can be white, green or brown in color. What are these? These are fungal patches.
Nutrition in unicellular organisms. Amoeba is a unicellular organism. It eats microscopic plants and animals that float in water. An amoeba ingests food by using its pseudopodia. When a food particle comes near an amoeba, it ingests this food particle by surrounding it with its pseudopodia. The pseudopodia close to form a small cavity called food vacuole. The ingested food particle is digested by the food vacuole. The food vacuole contains special enzymes which help in breaking down these food particles into a simple soluble form. This soluble food is absorbed by the protoplasm and assimilated to produce substances required for growth and development. The undigested food is thrown out by the contractile vacuole. Nutrition in multicellular organisms. Hydra, a multicellular aquatic animal, has tentacles which help it ingest its food. The tentacles help in catching and paralyzing small aquatic animals with the help of sting cells present in them. The food material is then ingested inside the body cavity through the mouth and digested. This digested food is absorbed by diffusion through the cavity walls and is assimilated in the cells of the body. It throws out wastes through its mouth.
Life cycle of silk moth. The life cycle of a silk moth consists of 4 stages. Stage 1. A female silk moth lays a number of eggs on mulberry leaves. Stage 2. The eggs hatch into tiny larvae called caterpillars or silkworms. Caterpillars eat mulberry leaves very quickly and grow in size. Stage 3. When a caterpillar is fully grown, it stops eating. It first weaves a net to hold itself. Then it swings its head from side to side. During these movements of the head, the caterpillar secretes a substance made of protein from its salivary glands. This proteinous substance hardens on coming in contact with air and becomes silk fiber. Soon the caterpillar completely covers itself in silk fiber. This covering is known as cocoon. Silk fiber is obtained from this cocoon. Inside the cocoon, the caterpillar changes into a pupa that slowly changes into an adult moth. Stage 4. When fully grown, the adult moth cuts the cocoon and flies out. Activity 1. To show that an object can be hot as well as cold at the same time. Take 3 beakers and label them A, B and C. Put some warm water in beaker A, normal tap water in beaker B and ice cold water in beaker C. Dip a finger of your right hand in A and left hand in C. After a few minutes, dip the fingers of both the hands in beaker B. What do you feel? You will find that the tap water feels hot to the finger of your left hand and cold to the finger of your right hand. Activity 2. To understand the process of conduction. Take a metal rod and fix it as shown in the figure. Drop wax on the metal rod at an equal distance using a burning candle. Now light a burner and place it under one end of the rod. What do you observe? The wax point 1 nearest to the burner melts first, then wax point 2, and so on. Activity 3. To understand the process of convection, take some water in a beaker, drop a few potassium permanganate crystals into it through a drinking straw so that the crystals settle only at the bottom and the water gets a little color. Do not stir the water. Now light a burner and heat this water. What do you observe? You will see that colored water rises up. Activity 4. To test the conductivity of air and water. A. Half fill a test tube with water. Drop a piece of wax wrapped in a wire mesh in it. This wax piece will sink to the bottom of the test tube. By holding the test tube in a tilted position, heat the upper part of the test tube as shown in figure A. Wax will not melt even when the water at the upper part of the tube starts boiling. B. Take a test tube of good quality, tilt it as shown in the figure B, and heat its bottom part. Insert your index finger into the mouth of the test tube. You will only feel a little heat.
Activity 1. Using litmus solution to test the nature of some substances. Collect red and blue litmus solutions or paper and the samples of various substances as lemon juice, vinegar, soap solution, baking soda, curd, etc. in separate test tube. Add a drop of these solutions on red litmus paper or alternatively add a drop of red litmus solution to all the test tubes. Record your observations of color changes in a table. Put a drop of these solutions on blue litmus paper or alternatively add a drop of blue litmus solution to all the test tubes. Record your observations of color changes in the table. Also write your conclusion from this activity. Activity 2. Using China rose extract to test the nature of some substances. Arrange a few China rose flowers, remove their petals and put these in boiling water. Let them soak in hot water for 10 minutes. Filter the solution. Your China rose extract is ready. What is the color of the extract? Keep 2 ml extract in one test tube as control. Do not add anything to it so as to match the track of color change. Test the rest of the solutions collected in previous activity with the extract and record your observations in a table. Activity 3. To neutralize the effect of acid, take a little amount of vinegar in a test tube. Add blue litmus to it. What color change do you observe? Now add baking soda solution drop by drop to the test tube. Does the color change to blue again? Count the number of drops of baking soda solution required to nullify or neutralize the effect of acid on litmus. Activity 4. To show heat is released during the neutralization reaction. Take 2 ml dilute NaOH solution in a test tube and add a drop of phenolphthalein solution to it. Note the color of NaOH solution and phenolphthalein solution. Are they colorless? What color change do you observe after addition of phenolphthalein? Does it change to pink? Count and add dilute
HCl.
drop by drop to the same test tube. How many drops does it take to change the pink color to colorless? Why does the color get decolorized? Add 1 drop of dilute NaOH to reaction mixture. Above solution. Does it turn pink again? Touch the test tube. Is it warm?
Is mixing of water and salt a physical change?
When we add salt to water,
The salt dissolves in water and water becomes salty, but no new substance is formed. It is just a mixture of salt in water in which the properties of both salt, salty taste, and water, quenching thirst, have been retained. Moreover, we can get back salt from the salt solution by the physical method of evaporation. If components of a mixture can be easily separated by a physical method, it is a physical change. Activity 2. To obtain sugar crystals from sugar solution, heat 1/4 cup of water in a pan. Add 1 cup sugar to it and dissolve by stirring. Keep on adding sugar till it is dissolved to prepare a saturated solution. Filter the solution in a beaker. Let the sugar solution cool down undisturbed, slowly at room temperature. You may put 2 or 3 crystals of sugar in the beaker. This is termed as seeding, as new crystals grow around them. Activity 3. To study the conditions required for rusting. Take 3 clean and dry test tubes and 3 iron nails cleaned with a sandpaper. Put 1 iron nail in each of the 3 test tubes. Add water to the first test tube such that the nail in it is half immersed in water. This arrangement will have nail, water, and oxygen, all 3 in contact. Add boiled water to the second test tube such that the nail in the tube is fully immersed in water. Put some oil on the top of water. By this arrangement, the nail would be in contact with water but without oxygen. By boiling the water, dissolved oxygen has already been expelled, and a layer of oil will stop the supply of oxygen to the test tube. Put some calcium chloride in the third test tube. This arrangement will be such that the nail would be in contact with oxygen, but not water or moisture. As calcium chloride is a dehydrating agent, it will absorb all the moisture present in the air inside the test tube. Leave the test tubes undisturbed for the reaction to occur. As the reaction is slow, it will take 2 to 3 days.
you will notice a brown coating around the nail put in the first test tube. There will be no change in the nails of second and third test tube. This is because the reaction has taken place only in the first test tube due to the presence of air and moisture. However, no reaction takes place in the second and the third test tube due to the absence of air and moisture respectively.
Activity 4. To show that a gas is formed during the process of chemical change. Take 1 teaspoon of baking soda, sodium hydrogen carbonate, in a beaker. Add water and stir well to make a solution. Now add some lemon juice or vinegar to it. What do you observe in the beaker? Does a gas come out with fizz? The same thing happens on opening a bottle of an aerated drink. This gas is carbon dioxide. Do you think that a chemical reaction has taken place? What makes you think so? Activity 1. To observe pressure exerted by air in all directions. Method. Clean an empty can and put some water in it. Boil the water for some time and then screw on the can cap. Carefully put this in a water bath and pour cold water on it. What do you observe? You will see that can crumbles from all sides. Reasons. In an empty can, air exerts pressure from inside as well as outside the can. The water vapor formed due to boiling drives the air outside to take its place, thus maintaining the pressure balance. But cold water condenses the water vapor and they settle down, creating a vacuum inside. This creates a pressure difference inside and outside the can, and this crushes the can from all sides. Activity 2. To demonstrate lift due to air pressure. What do you need? Piece of tissue paper. Method. 1. Cut a long strip of tissue paper and hold it in your hand. Leave one end free. You will notice that it hangs limp due to gravity. 2. Blow lightly above the tissue. What happens? It lifts up. Why? As the air above the tissue paper starts moving, its pressure drops. This exerts a force on the tissue, lifting it up. 3. Now blow harder. You will see that the harder you blow, the more the tissue paper is lifted. Activity 3. To observe behavior of air on heating. Method. Take 2 paper bags and tie them inverted with a stick as shown. Balance them and keep them in that position. Light a candle below one bag and observe carefully. You will notice the bag will start rising up. Reasons? The air between the bag and candle flame gets heated up and expands. This hot air becomes lighter and rises up to fill the inverted bag, causing it to move up. Tornadoes. A tornado is a violent spinning storm, typically shaped like a funnel with the narrow end on the ground. Tornadoes are one of the most deadly and fascinating forces of nature. They can also be described as a towering black funnel extending downward from the base of a large cloud. Tornadoes usually occur as part of a severe thunderstorm and are also called twisters. Tornadoes are usually formed over land. They are formed when there is warm, moist air near the ground and cold, dry air above it. The warm and moist air, on being heated by the sun, rises up. On rising, it cools and forms large clouds. To take the place of rising air, more and more surrounding air gets sucked into the bottom of the tornado. It also sucks in everything like trees, cars, houses, debris, etc. that comes near its base as it moves over an area. Activity 1. To study soil particles. Take some soil in a beaker, add water and shake it well. Let the beaker stand so that the soil settles down. What do you notice? You will notice that the heaviest components will sink to the bottom and the lighter ones will be on top at various levels, depending on how light the particles are. Try soil samples from different areas. You will find the relative proportion of the components is different in soil sample taken from different places. Activity 2. To study percolation rate of water in different soils. Take 3 similar plastic bottles. Empty mineral water bottles can be taken. Cut off top and bottom of all 3 bottles so that they look like hollow plastic pipes. Collect about 2 to 3 kilograms samples of 3 different types of soil. Now, place each pipe in the 3 soil samples in such a way that they are about 2 centimeters deep in the sample. Measure about 200 milliliters of water and pour it into the first pipe. Note down the time taken for the entire water to percolate into the soil. Repeat this with the other 2 samples. Now, calculate the percolation rate using the formula percolation rate
milliliters per minute
is equal to amount of water percolated
milliliters
by time taken in minutes. For example, if it takes 20 minutes for 200 milliliters of water to percolate, the rate of percolation is percolation rate is equal to 200 ml by 20 minutes is equal to 10 ml per minute. Activity 3. To test the presence of moisture in soil. Take a boiling tube and fill 1/4 of the tube with soil. Now heat it gently on a flame. You will notice some drops of water on the upper part of the tube. This shows that the soil contains water. The water vapor condenses after touching the cooler upper part of the tube. Now compare this heated soil with the original soil sample. Do you find any difference? Activity 4. To study absorption of water by soil. Take a filter paper, fold it and place it in a plastic funnel. Weigh 50 grams of dry powdered soil and put it in the funnel. Measure some water, say 100 ml, using a measuring cylinder and pour it drop by drop on the soil with a dropper. Pour water all over the soil and not at one spot. Keep pouring till water starts dripping. Subtract the amount of water left in the measuring cylinder from the amount of water you started with. This is the amount of water retained by the soil. You can find out the percentage of water absorbed by the soil sample with the following calculations. Weight of soil, 50 grams. Initial volume of water in the measuring cylinder, 100 milliliters. Final volume of water in the measuring cylinder, 60 milliliters, suppose. Then volume of water absorbed by the soil is equal to 100 minus 60 equal to 40 ml. Percentage of water absorbed is equal to volume of water absorbed by the soil by 50 into 100. That is equal to 40 by 50 into 100, which is equal to 80%. Activity 1. To prove that respiration produces heat. Take a little amount of vinegar in a test tube. What do you need? 2 test tubes fitted with thermometers and bean seeds. 1. In one test tube, put some actively germinating seeds, and in another test tube, put some boiled seeds. Note down the initial temperatures of both the tubes and leave the setup for a few hours. 2. After a few hours, record the temperatures shown by both the thermometers again. Observation. Temperature in the test tube containing germinating seeds will increase, while that in the second will remain the same. Conclusion. This shows that heat is generated during respiration. Inhalation, inspiration. When the ribcage moves up and the diaphragm relaxes, lungs expand. More space is created inside the lungs and we inhale oxygen-rich air. Exhalation, expiration. When the ribcage moves down and the diaphragm moves upwards, lungs contract. The space inside reduces, forcing the air to rush out, and we exhale carbon dioxide-rich air. Activity 2. To understand the mechanism of breathing. Cut a hole in the middle of a straw. Do not cut it through. Fit a small balloon at each end. Cement the ends and leave it to dry. When the cement has dried, bend the straw at the cut mark so that the hole points outwards. Make a V-shaped cut at the end of another straw. Fit the V-cut of the second straw into the hole of the bent straw. Cement the joint and leave to dry. Pierce a hole of the size of the diameter of the straw at the bottom of a transparent plastic cup. Keeping the plastic cup upside down, insert the open end of the straw into the hole from the inside. Cement the straw in the hole and leave to dry. Cut off the neck of a large balloon and carefully stretch it out as a sheet over the rim of the inverted cup. Pull down the balloon sheet and observe the small balloons. Push the balloon sheet up into the cup and observe the small balloons again. You will observe that when the balloon sheet is pulled down, the smaller balloons get inflated, and when it is pushed up into the cup, they get deflated.
Activity 1. To show that xylem conducts water in plants. What you need: flowering plants, beaker, water, red ink, microscope, knife. Method 1. Take a branch of a flowering plant and put it in a beaker containing water with some red ink. 2. Leave the branch immersed in water for half an hour. 3. Now cut a thin slice from the stem of the branch and observe it under a microscope. Observation: we can see only xylem cells stained with red color. Result: this shows that xylem transports water and minerals from roots. Activity 2. To show transpiration. What you need: potted plant, polythene bag. Method 1. Take a potted plant and water it sufficiently. 2. Cover a leafy branch with a polythene bag. 3. Keep the potted plant in sunlight for a few hours and observe. Observation: water drops in polythene bag will be observed as water loss from the leaves through the process of transpiration. Activity 3. To show that the phloem transports food material in plants. What you need: potted plant and a microscope. Method 1. Take a potted plant with thick stem. 2. Remove the phloem tissue from the stem. 3. Observe the stem after a few days. Observation: the stem shows swelling in the upper portion due to accumulation of food material. This is because the downward movement of food is blocked. Result: this shows that the phloem helps in the transportation of food from the leaves. The urinary system. Kidneys. Kidneys are small bean-shaped organs which filter blood to remove urea and other wastes along with excess water. Ureters. Ureters are tubes which pass the urine formed in the kidneys to the urinary bladder. Urinary bladder. The urinary bladder is a small, muscular bag-like organ which swells up when full of urine. It shrinks in size after urine is expelled out. Urethra. Urethra is the tube that allows the passage of urine. A healthy human being passes about 1.5 to 2.5 liters of urine every day. Activity 1. To study budding in yeast. Prepare a sugar solution and dissolve a pinch of dried yeast in it by shaking the beaker. Cover it with a lid and leave it aside undisturbed for an hour in a warm place. Put a drop of this solution over a glass slide and observe under a microscope. A small bulb-like protrusion is formed on the parent cell. This is called a bud or a daughter cell. The bud continues to grow until it separates from the parent cell, forming a new independent cell. Sometimes the buds may not separate from each other and appear as a chain. Activity 2. Take a potato. Cut out a small piece with an eye in it. Plant it in a pot with good moist soil. Observe it developing roots, shoots, and growing into a new plant. Cutting. In this method, portion of the plant containing leaf buds is cut from the parent plant. It is then induced to form roots and shoots by providing a favorable environment like moist soil with sufficient nutrients. Plants including banana, pineapple, rose, bougainvillea, sugarcane and cactus can be propagated by this method. Activity 3. To understand reproduction in plants by cutting. Take a leaf with some buds of bryophyllum. Plant these leaf cuttings in a pot. Observe how within a week these cuttings develop into new plants and resume flowering. Grafting. In this method, parts of one plant are fused with another plant. One plant is selected for its roots and is called the stalk. The other plant is selected for its stems, leaves, flowers or fruits and is called the scion. The scion gets essential nutrients from the stalk. The resultant plant possesses the features of both the grafted plants. Layering. It involves plants with long, flexible branches where a branch or a stem is still attached to the parent plant. It is placed in contact with moist soil, encouraging it to send out roots. Once the roots emerge, new plantlets are formed. These can be separated from the parent plant and grown as new individual plants. Grapes, raspberry, and jasmine can be easily propagated by layering. Tissue culture. Plant tissues have the ability to generate whole new plants. Living and actively multiplying tissue is obtained from the plant and is kept in a medium containing appropriate amount of nutrients. It is then treated with root and shoot-inducing chemicals in the laboratory. Once it develops into a plantlet, it can be transplanted into soil in the nursery and raised into a full-grown plant. Orchids, bananas, and chrysanthemums are grown using tissue culture. Tissue culture. Plant tissues have the ability to generate whole new plants. Living and actively multiplying tissue is obtained from the plant and is kept in a medium containing appropriate amount of nutrients. It is then treated with root and shoot-inducing chemicals in the laboratory. Once it develops into a plantlet, it can be transplanted into soil in the nursery and raised into a full-grown plant. Orchids, bananas, and chrysanthemums are grown using tissue culture.
Structure of a flower.
Pistil. Pistil or carpel, a female reproductive part, has a swollen lower part which is attached to the flower and is known as the ovary. An ovary may contain one or more ovules. Ovules contain female gametes. Gametes are reproductive cells. Carpel also has an upper part called stigma, where pollens are deposited, and stalk called style, which connects stigma and ovary. Stamen. It is the male reproductive part of a flower. The anther of the stamen contains very tiny, yellowish, spherical structures called pollen grains. Pollen grains are very light structures containing male gametes. Petals. These are the colorful parts of the flower. They help in attracting insects, birds, and other animals, which spread pollen and facilitate sexual reproduction. Sepals. The outermost holes of a flower are called sepals. Cross-pollination. In cross-pollination, pollen grains are delivered by pollinators to the stigma of a flower or a different flower on the same plant. Apples, pears, and plums show cross-pollination. Self-pollination. In self-pollination, pollen grains fall on the stigma of the same flower. Self-pollination is seen in soya beans and peaches. Fertilization in a flower. When the pollen grains land on a stigma, the pollen begins to form a pollen tube. The tube grows along the wall of the style and curls at the bottom of the ovary. The tube breaks through an opening in the ovule to reach the female gamete egg cell. The male gamete moves down the tube and fertilizes the female gamete, forming a zygote. Activity 2. To study the characteristics of a pendulum. What you need: an inelastic string, a small metal bob. Method: 1. Take a thread with a bob tied to its one end. Tie its other end to a fixed point. 2. Give a slight push to the bob. The bob will start oscillating. This arrangement is called a pendulum. 3. Using a stopwatch, see how much time the pendulum takes to complete one oscillation. Record it. Now stop the bob and let it oscillate after displacing it by a smaller distance. Again, record the time taken to complete one oscillation. Observation. You will notice that the time of oscillation remains the same in every situation, whatever is the length of the arc made by the oscillating pendulum. Conclusion. In every position, a pendulum takes equal time to complete one oscillation as long as the length of the string remains constant. Series circuit. A circuit where two or more bulbs are connected one after the other in a series is called a series circuit. In a series circuit, the complete circuit should be properly connected with no break or disconnected components in between. It does not give any output if the circuit is broken at any point. In this circuit, as in decorative lights, even if one bulb fuses, the entire series of bulbs stop glowing as the flow of current disrupts. Series circuit. A circuit where two or more bulbs are connected one after the other in a series is called a series circuit. In a series circuit, The complete circuit should be properly connected with no break or disconnected components in between. It does not give any output if the circuit is broken at any point. In this circuit, as in decorative lights, even if one bulb fuses, the entire series of bulbs stop glowing as the flow of current disrupts. Parallel circuit. When two or more bulbs or appliances are placed in such a way that each comprises a simple circuit placed parallel to each other, the arrangement is called a parallel circuit. In this circuit, even if one bulb blows a fuse or an appliance stop working, the rest of the circuit will remain operational. Activity 1. To demonstrate the magnetic effect of electric current. What you need: a magnetic compass, a bulb in a bulb holder,
A 6-volt battery.
a switch, a plastic box, a screwdriver, some insulated copper wire. Method 1. Make a circuit using battery, switch and the bulb in the holder. Put the switch on. Check whether the bulb lights up or not. Now turn the switch off. 2. Place the magnetic compass on the wire and ensure the needle points towards north. Move the wire so that it points north-south in line with the needle. 3. Put the switch on. Does the bulb light? Now check what happens to the compass needle when electricity flows through the wire. Turn the switch off. See what happens. Try this with the battery connected the other way round. Observation. The north tip of the compass needle points in the direction of current. Conclusion. An electric current produces magnetic effects around a Activity 2. To make an electromagnet. What do you need?
4.5-volt battery.
A pencil, a large iron nail, insulated copper wire, a compass needle, and safety pins. Method 1. Wind the wire tightly and uniformly around the pencil. 2. Connect both ends of the battery to the wire. This electromagnet will be strong enough to affect a compass needle, but too weak to attract anything. 3. Replace pencil with iron nail. Now you will have an electromagnet strong enough to attract safety pins. Activity 3. To detect the strength of a magnet using a magnetic compass. What do you need? 2 iron nails, compass needle, batteries, copper wire. Method. Case 1. Take 2 iron nails and wind the copper wire as shown in the figure above. In case 1b, the nail has more turns of the wire and the magnetic compass needle shows more deflection. Case 2. Now increase the number of batteries in case
2b.
The compass continues to show more deflection in case 2b. Working of an electric bell. When the switch is pressed, the circuit is completed. The current flows through the wire and the iron rod becomes an electromagnet. The tiny iron hammer is attached by the electromagnet and strikes on the bell. As the hammer moves towards the bell, the circuit is broken and electricity stops flowing through it, and so the iron rod loses its magnetism. As the rod loses its magnetism, the hammer returns to its original place. The circuit makes fresh contact once again. Once again, electricity flows through the circuit and the rod acquires magnetism, and so the hammer strikes again. The cycle repeats itself and the bell continues to ring as long as the switch is kept pressed. Phenomena occurring due to rectilinear propagation of light. Shadow. The shadow of an opaque object is formed only when it comes in the path of light, day and night. When the sun rays fall on a particular portion of the earth, that part of the earth experiences day and the rest of the planet experiences night. Pinhole camera. The image in a pinhole camera forms only due to rectilinear propagation of light. Activity 1. To observe reflection of light by polished surface. What you need? A plain mirror. Method 1. Stand in a dark room with only a small part of a window open to let light enter the room. 2. Place the plain mirror in the light. Observation. No light is seen behind the mirror. A patch of light is formed on the table in opposite wall. 3. Turn the mirror slowly in one direction. Observation. The patch of light on the wall also moves. Conclusion. A plain mirror or a polished surface reflects the light. Activity 2. To observe image formation in a concave mirror. What you need? A concave mirror, a candle, a white screen and a mirror stand. Method 1. Place a lighted candle on a flat surface in a dark room. Also place a white screen and a concave mirror in line with the candle. 2. Place the mirror at a large distance from the candle, say 50 cm. Move screen away from the mirror. Observation. At one position of the screen, an inverted image of the candle flame forms on the screen. 3. Take the mirror towards candle, say 10 cm closer. Observation. The image disappears from the screen. 4. Again move the screen back to get an image. Observation. The size of the image obtained increases. 5. Continue the process. At one position, no image will be obtained on the screen. However, an erect enlarged image can be seen in the mirror. This will be a virtual image. Activity 3. To observe image formation in convex lens. What you need? A convex lens, a candle, a white paper or screen and a lens stand. Method 1. Place a lighted candle on a flat surface in a dark room. Also place a white screen on one side and a convex lens between the candle and the screen. 2. Place the lens at a large distance from the candle, say 50 cm. Move screen away from the lens. Observation. At one position of the screen, an inverted image of the candle flame forms on it. 3. Move the lens towards the candle, say 10 cm. Observation. The image disappears from the screen. 4. Again move the screen back to get an image. Observation. Size of the image obtained increases. 5. Continue the process. At one position, no image will be obtained on the screen. However, an erect and enlarged image can be seen on the same side as the candle. It is a virtual image.
Activity 4.
Cut a circle of 15 cm radius out of thick white card paper. Draw 7 equal segments on it. Color the segments using the color violet, indigo, blue, green, yellow, orange and red respectively. Pierce a small hole in the center and pass the pointed end of a pencil through it. Hold the pencil horizontally so that the card is vertical. Tap the side of the card so that it rotates fast. Note the color of the disc. What do you observe? The disc appears grayish-white and you do not see the 7 colors. Water cycle. Due to heat of the sun, water of seas, oceans, rivers, lakes, etc. gets evaporated continuously. Transpiration of plants also adds water vapor to air. This water vapor rises and condenses to form clouds. When clouds are saturated with water vapor and become heavy, precipitation occurs which accounts for rain. Thus, water falls back to the earth. This constitutes the water cycle.